Hello. Today we are presenting the journal article Virus-Based Vectors, a new approach for the production of recombinant proteins. This journal comes from the Journal of Applied Biotechnology Reports, and it was published in March 15th of 2019. So the importance of this study is to analyze the novel use of viral-based vectors to introduce the gene of interest to plant hosts by transient expression to basically produce a recombinant protein through heterologous gene expression. Um, they study first generation and second generation as well as analyze biosafety considerations. To preface this presentation, we're going to start off with recombinant proteins. So recombinant proteins are proteins that have been genetically engineered through genetically en- sorry, excuse me, disregard that. Recombinant proteins are produced when scientists genetically engineer vectors with using recombinant DNA. They basically insert recombinant DNA into a vector, and then that vector is then transformed into a host expression system where that host expression system will then take that vector and start expressing it through central dogma and express your protein of interest. Depending on your host expression system, it will either be transformed or transfected, and through central dogma, the DNA will go transcribe into mRNA, and then that mRNA will then be translated into your protein of interest. So biotechnology is built upon recombinant proteins, and to get these recombinant proteins, you need host expression systems. So what kind of host expression systems are you guys familiar with? Cell cells, bacteria, yeast, transgenic animals. Cool. Seems like you guys are familiar. You guys been paying attention in class. So, one of the most popular used in the industry is bacteria, specifically E. coli, with it being gram negative. It is one of the most used host systems for recombinant protein production. It is relatively cheap and it has a very fast doubling time, making it very ideal to culture and get a large yield of proteins, but it does lack proper post translational modifications, and it expresses more simple proteins. And a consideration with this host expression host is that it has inclusion bodies, codon bias, and endotoxin issues, which is an issue for FDA compliance and producing your product. Another popular expression host are mammalian cells, specifically CHO cells, Chinese hamster ovary cells. They are able to express larger and more complex proteins as well as provide those post translational modifications such as glycosylation of your protein. But the thing is, they have a slower doubling time and there is a higher cost when it comes to them due to media. But they are able to also secrete the protein, which makes the downstream processing a lot more easier. Another expression host system that is used in the industry is Pichia pastoris and Saccharomyces cerevisiae, which is yeast. um, they have a rapid growth, so they have a pretty fast doubling time, but not as fast as bacteria cells. They have easy genetic manipulation, which is ideal for being able to manipulate and use it to express your protein of interest, and they're pretty cost effective. Um, Another expression host system that is also recognized are insect cells. They're similar to mammalian protein processing and are able to be in static or suspended cultures. But with insect cells, they have more demanding culture conditions, which can pose as a consideration if when trying to culture, as they may be more sensitive and they may be more susceptible to not working as well as compared to other expression host systems. Two other expression host systems that are recognized are transgenic animals, which is a more novel approach to expressing proteins. They have high levels of expressions of their protein of interest and have complex post translational modifications, and the proteins are actually expressed in their native forms. But since it's so novel, it has a poor regulatory record with the FDA as of right now, and they're also are more costly as you are dealing with a whole animal rather than just a unicellular. And as well, with it being a transgenic animal, it has a longer timeline, which can take about months to years, depending on which animal you're using. And with transgenic animals, there's also transgenic plants, which are low cost and have low risk of contamination with animal pathogens since it is a plant cell. They are easy to scale up, have stability, and they do have the presence of metabolites, but are also able to produce N-glycosylated proteins. And pesticides, herbicides, and toxic plant metabolites are a consideration for contamination. So, plants as expression systems. This was one of the main points that the journal touches upon. So, Agrobacterium tumefaciens is a gram-negative soil-borne bacteria that is used to infect plant cells. Using plants as an expression system has a high production of the protein of interest. Specifically, in this presentation, we're going to be focusing on transient expression by viral vectors using Agrobacterium tumefaciens. With this approach, transient expression can occur in about 2 to 7 days using this approach with Agrobacterium tumefaciens containing your gene of interest. With this is a low cost and speedy process, and it shows an efficient transfer of your genes of interest to your plant host system. And as well, with this approach, they do require the least amount of water, CO2, and minerals. So, Agrobacterium tumefaciens, as I mentioned before, it was a gram-negative bacteria that is soil-borne, so it is more familiar with the natural habitat of plants. It is a tumor-inducing bacteria that infects plants, meaning that it does cause tumors. So, naturally, Agrobacterium contains this tumor-inducing plasmid. Scientists have utilized that TI plasmid, tumor-inducing, to insert their gene of interest and use it as a vector to facilitate producing proteins to get the tDNA into your host cell. So, Agrobacterium is cultured with your gene of interest with this TI plasmid incorporated into Agrobacterium, and it is cultured in suspension. With the culture, you then introduce it to the host by spraying, injecting, or using vacuum infiltration, and the gene of interest will then be transfected into the plant through agrobacterium. Agrobacterium basically facilitates introducing the gene of interest to your plant. So, to compare and contrast transient and transgenic expression systems, transient expression is more of a short term approach. The genes don't incorporate into the chromosomes of your said cell, and it won't be able to pass it on to future generations. So it is a very temporary approach, but the yield of your protein is larger up front as the plant allocates their metabolic resources to produce your protein of interest. So this timeline can take about days to weeks, and the yield for protein is about 1 to 5 grams per kilogram of protein produced per plant biomass, as opposed to a transgenic approach. It is more long term since the genes are being integrated into your host's genome. So that means that the genes will be passed on from generations to generations, making it more stable for your protein production. But this does take about a longer time. It takes about like 6 months. But you will have a consistent and more stable protein production, which can pose as a long-term higher yield protein production rather than having a high production of protein up front. So this has a lower protein production up front in the transgenic aspect. So we've been talking about using RNA viral-based vectors. The first time in viral RNA expression has occurred in vivo and in vitro was in 1984. They utilized the TI plasmid, tumor-inducing plasmid, which is found in agrobacterium as stated before. This tDNA is transferred from agrobacterium to the plant cells, and it contains viral RNA. So the TI plasmid consists of the origin of replication, the virulence region, and as well as the tDNA region, which is flanked by the left border and right border to mark the tDNA region. The virulence region is the viral part of the vector of the plasmid, that facilitates cell-to-cell movement, systemic movement, and the origin of replication is so that it replicates. These RNA viral-based vectors pose flexibility, scalability, and safety for high production of your protein of interest, which is encoded into the tDNA region. So, viral RNA strands that are mainly and commonly used in viral-based vectors, specifically for plant expression, is tobacco mosaic virus (TMV), potato virus X (PVX), cowpea mosaic virus (CMV), and tobacco rattle-virus (TRV). These are constructed into your viral-based vectors, and these vectors will then go into your agrobacterium, and then the agrobacterium will facilitate the plasmids into your plant to incorporate the tDNA, which will then facilitate your protein production. So to get that agrobacterium to be introduced to the plant, you introduce a culture, your suspended culture of agrobacterium to your plant. So as I mentioned before, you either introduce it via spraying, injection, high pressure spraying, injection, or vacuum infiltration, commonly used in the first generation of viral based vectors, were spray and injection. But as technology and more scientific findings were found, vacuum infiltration posed to be the most efficient and cost effective for introducing agrobacterium to your plant leaf. So, spraying, high pressure spraying, you're using high pressure spraying. Injection, you take a syringe and inject it into your plant appendage, plant leaf, whatever plant. And vacuum infiltration is where the plant tissue will be submerged into a vessel containing your agrobacterium culture. So it will be fully submerged, the plant tissue. And then within this vessel, there will be an increase and decrease of pressure to ensure that agrobacterium is fully facilitated all over your plant so that it covers all of your plant tissue. And with vacuum infiltration, it replaces the virus's function, the primary function of primary infection and systemic movement, meaning that there will be less reliance on the virus itself to be incorporated through cell to cell movement and systemic movement, but rather you're already forcing it to move into the cells rather than just relying on it. This approach is very speedy and it has high expression levels and has proper function of the virus. Excuse me. And up to 5 grams per kilogram of plant protein is obtained per plant biomass. Up to 5 grams per kilogram protein obtained per plant biomass. Okay, so now we're going to talk about transient expression in viral vectors mediated through agrobacterium. So, as you can see, the tDNA is facilitated through the agrobacterium. The agrobacterium will either sense plant signals, plant injury signals, or synthetic plant injury signals, and that will basically facilitate the tDNA that is in your plasmid. So the tDNA will move into the plant cell and it will enter the cytoplasm. From the cytoplasm, it will be incorporated into the nucleus, where the tDNA will be transcribed. It will be transcripted, It will be transcripted into mRNA. So from the mRNA, it will move outside of the nucleus and go back into the cytoplasm, where this RNA will be transcripted into mRNA and RDRP. So from the nucleus, the mRNA will be translated in the cytoplasm into RDRP, and then these RDRPs are going to take minus RNA strands from your transcription and produce minus RNA. Okay, so from this minus RNA, there will be 2 pathway options. So one pathway option is that the mRNA that has differential size specific to your viral protein and target protein will be translated into your protein of interest, coat proteins, and TGB, which stands for triple gene block, which is used for cell-to-cell movement. And so you have your protein of interest, TGB, and the coproteins produced, which are your protein products. And in the second pathway option, your minus RNA will be fully transcribed into a full viral RNA from the M minus RNA. And then this complete viral RNA will bind to your TGB, which facilitates cell-to-cell movement. And that TGB will move to the next cell to incorporate your viral RNA into the next cell's cytoplasm to continue this transient expression and repeat the loop. So basically, it's encoding for copies to make more proteins and as well as more code to make the code that makes the proteins that makes the code, if you pick up what I'm trying to say. Okay. So now that we've got that down, first generation, which is when they first started testing out using viral-based vectors. So with this approach, they used a full virus strategy, which expresses your gene of interest in addition to the own natural genes of the virus. So it would have your gene of interest as well as the virus in the plasmid. And to introduce your tDNA, you would have to use agrobacterium and as well as using the tDNA and spraying it on your plants. They would do high pressure spraying on top of the plants to try to incorporate it. But with this approach, scientists weren't able to get bottoms of the leaves, so there wasn't a full saturation of your plant with your tDNA and agrobacterium. Host plants were transformed by the virus suppressor factor, and your gene of interest was essentially expressed by the promoter and viral promoter that was included. And as well as the gene of interest is sent as a transcript of infecting DNA or mature viral particles. And this whole process of infecting the whole plant took about 2 to 3 weeks. And there was a relatively low protein yield, as it was 0.04 to 0.3. So it wasn't really as optimized as it is right now. But to note, with this first generation, there was successful expressions of the immunogenic epitopes, which were binded to your capsid protein of the virus. So this basically means that the immunization of the produced epitopes, disregard that. I didn't. So this was a successful representation of the tDNA being expressed and expressing your protein of interest. So essentially this means your process was working. With the immunization of the produced epitopes, it showed a special degree of protection in animal models. So this means that it's working. And throughout this approach, there was no recorded cases of contamination from mammalian-based viruses, which is pretty good, pretty positive, pretty good, especially for the application of pharmaceuticals for humans. That means that it's relatively safe from mammalian-based contaminants such as the mammalian-based viruses. So you don't have as big of a worry of contaminants from that. But this approach does require a transfection of a large number of plants to get a high protein yield, as there is a low protein yield per plant. Some limitations with this approach, though, is that you are limited to having to infect. You need to injure the plant tissue to infect the plant with your tDNA and the agrobacterium, which poses as a concern for contaminants to get in as you're having to wound the plant, and as well as the plant having to spend metabolic resources to focus on fixing that wound. Another limitation that was found were the viral particle assembly, which affects the effectiveness and adequacy of the whole process of producing your protein, as your viral particles aren't assembling, so you're not having as many workers per se to produce your protein of interest. Another limitation was the insert size of your gene of interest. If it exceeded a certain amount, it would be destroyed and your protein of interest would not be able to be expressed since of the large size. So you're limited to a smaller range of genes and proteins. As well as this caveat, there was a bias to the expression of the capsid protein of the virus. So rather than producing your protein of interest, there was also production of the capsid protein, and it took a bias towards so meaning that it allocated more resources to producing the capsid protein rather than your protein of interest, which resulted in a lower yield. It is also systemic movement specific, which is a reliance on this viral aspect of systemic movement from cell to cell. And another notable limitation that was found was that since it can be recognized as a virus within mammalian hosts, it can trigger an immune response, which is not ideal to what you're trying to achieve with pharmaceuticals, as you're trying to have no hiccups. But as science and technologies persisted, they continued to improve, and now we have second generation of viral-based vectors, which is deconstructed viruses. So in this approach, only essential viral fragments are incorporated into the vector, and these viral fragments are essential for gene expression. And you're removing the capsid proteins of the vector, which replaces systemic movement ability by agrobacterium and tDNA. So you would have to incorporate agrobacterium and your tDNA through agroinfection. But to compensate for required functions that were removed from your vector, they were provided by non-viral derived sequences of the vector. And this modified vector resulted in DNA processing in above 93% of cells, which is 130 times better than non-modified vectors, as the journal stated. So this was a highly efficient approach to DNA processing and improving the process of transient expression. Some notable characteristics regarding second generation was that it prevents the production of infectious viral particles, which is solid. So your host isn't recognizing it as an infectious viral disease or whatever and causing an immune response. And in the second generation, it assists gene expression Also, assembly of viral elements allow high expressions of your gene of interest, which then in turn causes a higher yield of your protein as more of your gene of interest is being expressed. And since you lack that capsid protein, you have more room to be able to insert genes, specifically larger than 2 to 3 kilobases, and proteins with a molecular weight higher than 80 kilodaltons are able to be expressed. Drink some water real quick. Oh my god, I'm at fucking 30 minutes. Hold on. Holy shit. Excuse me. Okay, let me stop because I gotta go to sleep. Thank you.